Hi, Science 9 students. I'd like to welcome you all to this session. This is going to be a review session for our second test, which is going to be on December 2nd. And again, I just want to remind you that the two ways to review for this test, assuming that you've already done lessons 11 to, starting at 7 to 11, and then also lessons 12 and 13 have review material on them, and they're on the second part of the lessons. So what I would focus on is the review material, knowing the material, and doing the questions on lessons 12 and 13, and also knowing the questions and material on this whiteboard session. And if you know all that material, you should do well on the test that's on December 2nd. Okay, good stuff. So here we'll go. We'll get started then. And so first of all, you can see here that we've got a little diagram that's talking about pH. And as you know, pH numbers always go from 1 to 14, or 0 to 14, I should say. 0 being the most strong acid, and 14 being the strongest base. Okay, so numbers that are in between, or pH numbers, are neutral. And so, as we can see, we have strong acids, which are very low or close to 0. And as we get close to 7, but still lower than 7, we have weak acids. Okay? And then strong bases are going to be up near 14, somewhere 12, 13, 14. And weak bases are going to be just a little bit above 7. Okay. So we know that acids and bases neutralize one another in a neutralization reaction. And do you know why it is that people use medicines like Tums? Well, Tums is used to treat a stomach that has too much acid in it. So can you guess what type of chemical is found in Tums? Well, of course, it's going to have a base in it, right? Because acids and bases neutralize one another. So a base will neutralize or cancel out an acid, and an acid will neutralize a base. So how does that work exactly? Well, you've got an acid that has a low pH. It's being combined with a base that has a high pH. And when you combine them together, you get water. You also get salt as well, but it gives you a neutral pH. Okay, so moving on to the next page, remember this is the overall neutralization reaction when acids are added to bases. So you're adding acid plus base, and you're getting salt plus water. And again, you're going to end up with a neutral pH of 7. So let's check out the first question here. First question says, the graph below shows what the pH looks like in someone's stomach after taking larger and larger amounts of Tums, which is a base. And the question says, what amount of base, in other words, Tums medicine, would represent the correct amount to take in order to relieve a stomach acid problem? Okay, so we're starting off with a low pH when we have a stomach acid problem. We're adding greater and greater amounts of base, and we're asking ourselves the question, what's the correct amount of base? As we can see, when we add more and more base, the pH is going up, right? That makes sense. Well, what the correct amount is going to be is a pH of around 7. So that's in between 6 and 8, right? Because that's a correct amount because we want like a neutral pH, right? A pH like the pH of water. So we would say that this amount of base is going to be the correct amount of base. So we can see the 10 grams of base gives us a neutral pH, and so that's the one we would choose. Okay, so the question says what amount of base are we going to use? And you can see that we would use 10 grams of base. So the answer would be C. Okay, good stuff. So let's check out the next question, or the next amount of, the next area of focus, I should say. So I'm just talking here about common acids and common bases and common salts. So those are kind of like everyday ones that we see a lot of the time. So things like vinegar, that's a common acid. Another thing is hydrochloric acid. That's found in your stomach. We also call it HCl. And you can see that acids are going to have H in them, or hydrogen. Bases, on the other hand, are going to be different. They're going to have OH, which is called hydroxide. And some things like baking soda that you have around the house, that's going to be an example of a base. Ammonia is a very strong base. Sodium hydroxide is also a really common base. And again, we can see that they always have, or often have, OH. Of course, you probably know when you add a base like baking soda together with an acid like vinegar, something happens, right? You get a lot of bubbling and stuff going on, and you get a neutralization reaction. Okay, so again, common acids have H, bases, many of them have OH, and salts, we're familiar with sodium chloride, that's table salt, another one is calcium chloride. Alright, so we have a question here where we're needing to add, or rather match, a definition for these different substances with the name of it. Okay, so think about a base. What did bases always have in them? Well, remember, they always had an OH, which is called hydroxide. So the answer that we would put here would be substance 3, calcium hydroxide. As far as an acid, remember acids like HCl, they have H in them. So hydrochloric solution, that's just kind of another way of saying hydrochloric acid, right? So for this one, we would write substance 2. H2O, of course, we know is water, substance 1. And salt, calcium chloride would be one example, and we're probably more familiar with sodium chloride. Okay, so moving along, we can see these strips here. These are called litmus papers, or litmus strips. You might have at one point been in a pool, and someone comes along and they stick a strip like this in the pool, or maybe they use another sort of testing system. But these things test the pH and let us know how acidic or how basic the pool is. So blue litmus papers change color when you add them to acids. They stay the same color if the pH is neutral or basic. And again, they change if you have an acid. Red litmus papers change color when you add bases to them. Okay, now if you want to remember, there's a technique to know that will help you so you don't even need to know what exactly blue litmus does or red litmus does. All you need to know is that blue equals base. Okay, and again, they both have blue in them, so that kind of helps. So in other words, if you have blue litmus and it's blue, you know you have a base. If it changes to pink, you know you have an acid. Same with red litmus. If it's blue, you know you have a base. If it's red, you know, or pink, you know you have an acid. Okay, so just remember blue equals base. And so we've got a question here that kind of taps into that. It says, which of the following test results indicate a base? First, if red litmus paper changes color, well, that makes sense, doesn't it? Because red litmus is going to change to blue, so that would be a base. B says if red litmus stays the same color, no, because we know the base has to be blue. If blue litmus changes color, no, because we know that it's got to be blue for a base. And if pH changes color, okay, so we know that the answer for this one would be A. If red litmus changes color to blue, then we know we have a base. Okay, good stuff. Now moving along, question 4 says, if a strong acid was spilled, which of the following would be the best product to clean it up? Well, what kind of pH does a strong acid have? Okay, of course we know acids are below 7, and we know that the lower you go, the stronger the acid is. So, if you want to clean up a strong acid, you need a strong base. So instead of having a really low pH, you're going to have a really high pH. So we're going to choose the one with the highest pH here. That's going to be ammonia, pH 11. All right, next question. These pH levels were taken at a campsite. What conclusion would you draw about what's happening over time? Okay, so we'll focus on site 1. We see before, during, and after. So that's the change that's happening over time. We can see the numbers are going down, aren't they? The pH values. So that means, of course, that it's becoming more acidic. We see the same thing with site 2, don't we? The numbers are going down, more acidic. So what conclusion are we going to draw about the campsite? The soil is becoming more acidic. All right, that wasn't too bad, eh? So on to the next page. It says, when a pool manager tested the water with red litmus, it turned blue. In order to neutralize the water in the pool manager should add. Okay, so before we correct the problem and see what we need to add, we need to see what we actually have. So when we tested the water with red litmus, it turned blue. So if you check out here, red litmus turns blue when we have a base. And we already knew that before, right, from the last question. So we know that we have a base, and we need to correct that problem with something else. Okay, what are we going to have to add to that base to correct the problem? Well, of course, an acid, right? So we're going to choose the answer that has acid, and that's going to be hydrochloric acid diluted. Okay, so again, find out what you have using the chart, and then find out what's the opposite to correct that problem. If you have an acid, you need to add a base. If you have a base, you need to add an acid. Okay, good stuff. So here's something that's kind of interesting. When you decrease the pH, it actually becomes 10 times more acidic. Or if you increase the pH, a solution becomes 10 times more basic. Okay, so when you go from pH 2 to pH 1, it's becoming 10 times more acidic. But when you go from pH 3 to pH 1, it's becoming 10 times 10, in other words, 100 times more acidic. Okay, so let's check out the next question then. It says sour milk has a pH of 5. A particular brand of toothpaste has a pH of 8. How many times more acidic is milk when compared to toothpaste? So what we need to do is figure out how many times the number is increasing. So we're going from 5 to 6, 6 to 7, 7 to 8. Okay? But we're comparing how many more times the sour milk with pH 5, how many more times acidic it is than toothpaste with a pH of 8. Okay, so if we go from 8 to 7, that's 10 times more acidic. If we go from 8 to 6, that's 10 times 10, or 100 times more acidic. And if we go from 8 to 5, that's 10 times 10 times 10, which is 1,000 times more acidic. So the answer would be D. Okay. So at this point, we're going to look at the difference between organic and inorganic substances. So if you look at these examples, this is methane, and this is methanol. These are organic substances. The reason why they are, just look at their formulas actually, and see if you can figure out what they have in common. What elements do you see in both these substances? Well, you see C, which is carbon, and you see H, which is hydrogen. So all organic substances have carbon in them, and most of them also have hydrogen as well. And remember, organic substances are found only in living things. Now, inorganic substances do not have carbon in them. So they can be found in both living things and non-living things. And H2S is an example of an inorganic substance because it doesn't have carbon in it. All right, so here's a question. State whether NaCl is an organic or an inorganic substance, and explain your reasoning. Okay, well, because it doesn't have carbon in it, it's got to be an inorganic substance. See, this is actually Cl, it stands for chlorine rather than carbon. Okay, so no carbon, then it's not an organic substance. All right. So what we're going to talk about now is what happens in the death of a lake. All right, so what causes a lake to die? Well, what happens is, first of all, actually adding something like fertilizer is going to cause the eventual death of a lake, even though fertilizer has lots of substances that are nutritious, you could say, for organisms in the lake. But what happens is you start off, a fertilizer adds phosphorus and nitrogen, P and N, the elements that have P and N as a symbol for them. Okay, what's going to happen then is those two substances, P and N, are going to cause algae to grow. Now, algae is kind of like a plant in some ways. It carries out photosynthesis just like any plant. And so when photosynthesis takes place, we know that one of the things it produces is oxygen. So in the beginning, oxygen levels are going to be high because of all the algae producing all of that oxygen during photosynthesis. What happens though is eventually that algae, after it grows, it's going to die. And when it dies, bacteria is going to feed off it. And as it feeds off it, eventually the bacteria are going to use up a lot of the oxygen that's in the water. That becomes a problem then. If you have low oxygen levels, creatures like fish that are living in the water are going to die. So those are all the steps that are taking place. And if you think about them one after the other, they all kind of logically connect. So let's look at a question that taps into this stuff that we've just been discussing. So question 9 says, what is the cause of each of the following? So the first thing, the growth of algae. Well, that's because we're adding fertilizer, which has phosphorus, or P, and nitrogen, or N. Okay, B, what's the cause of the initial high oxygen levels? Well, again, we know that algae produces oxygen during photosynthesis. Now, what causes the death of the fish eventually? Well, we knew that the algae, once they die, are going to be fed on by bacteria, and the bacteria are going to use up all the oxygen, or a lot of the oxygen in the water. And so there's not enough for the fish. And that's what happens next. Okay, so we've got another question here. It says the dissolved oxygen concentration was measured in 2 different lakes over time. One of the lakes had large amounts of fertilizer added to the lake. Okay, so this is kind of similar to what we were talking about before. It says decide which lake had fertilizer added to it and choose the correct reason to support the choice you've made. Okay, well, we know that what's going to happen is when we add fertilizer, first of all, algae is going to grow, and we're going to have high oxygen levels because of photosynthesis that's taking place. So we expect the oxygen levels to be high in the beginning. So that kind of looks like graph 2. Because what happens next is that the algae is going to die, the bacteria are going to feed off it, bacteria are going to use up all the oxygen, and then we're going to have low oxygen levels. So this graph makes sense based on what we talked about the series of steps before. So we know it's going to be lake 2, so it's either answer B or D. And is it going to be bacteria growth followed by algae growth, or algal growth followed by bacterial growth? Well, that's going to be the one, right? Because first of all, the algae growth, the algae grow because of the fertilizer, and then eventually they die, and bacteria feed on them. So it's going to be answer D. Okay. Now, in water, we can have 2 different types of indicators. When we were talking about pH paper, litmus paper, that was a chemical indicator. But a biological indicator is some sort of creature that's growing in the water that indicates something about the water. Now, there's a difference between microbiological indicators and aquatic invertebrate indicators. Microbiological indicators are bacteria that can indicate health concerns. E. coli is one of the most common bacteria when there's a health concern. So if you get a lot of E. coli growing in the water, it could indicate a really serious health problem. Whereas if you get these other guys, like for instance a water strider growing in the water, it doesn't necessarily mean there's a problem with the water. It just means there's something really specific about the water that we can assume or infer because of the presence of those guys in the water. So for instance, one of these guys might indicate that the water has a certain amount of warmth because those guys only grow when it's a certain temperature. Some of these other guys might only grow when the pH is a certain pH. So these involve things like insects, shrimp, and other creatures that have no backbone. That's why they're called invertebrate indicators. Okay, so again, those guys grow when there's a certain temperature or a certain amount of acidity, and they don't represent pollution, whereas the bacteria does. Okay, so we got a question here. Jim tests the water in 2 lakes and finds E. coli in one lake and finds a dragonfly nymph in another lake. So question A says, which of the organisms is an aquatic indicator? Well, that would be the dragonfly nymph. Second question says, what conclusions might Jim draw based on the organisms found in each of the lakes? Well, we know that if he finds the E. coli bacteria, that means there's a problem, there's pollution. Whereas the dragonfly nymph does not mean that. It just means that there's a certain pH, maybe it's a low one, maybe it's a high one, or a certain temperature, again, maybe lower, maybe high, and that's all it tells you. Okay, right on. So let's look at the next question. It says, some tropical fish prefer an environment that is slightly acidic. Based on this info, what do you think would be the most likely concentration of these fish? So we need to take the info that we have. We're told that the pH is going to be slightly acidic, and we need to find a concentration based on that. Well, what is slightly acidic? Well, neutral is 7, so slightly acidic would be something like 6. So if we take 6 and draw a line out to the graph, and then draw a line down, we would get a concentration of about 20. And that's going to be our answer. The answers were 15, 20, 25, and 30, and so we're going to choose the answer of 20. Okay. So, moving along, next question says, some organisms are more sensitive to changes in the pH of their environment. For these organisms, the more acidic the environment becomes, the more the number of organisms is reduced. Okay, so when you talk about a sensitive creature to pH, first of all, let's talk about a creature that's not sensitive to pH. If a creature is not sensitive to pH, that means it's not affected by pH. So if it's not affected by pH, if the pH changes from somewhere between 2 to 8, then the numbers are going to stay the same. You can see this creature, number 1, is at this level of numbers, and it's not really going up or down much, depending on the pH. Same with this creature. This one's a little more sensitive, but you can see that this creature, number 3, is really sensitive, because as the pH goes down, its numbers go down. Alright, so that's how you determine what kind of creatures are sensitive to pH, whether their numbers go down. And again, if their numbers stay the same, then they're not sensitive to pH because they're not affected by it. Alright, so at this point we're going to talk about something called biomagnification. It's a big word there. Now, what do you think is being magnified when we're talking about biomagnification? Well, what it is poisons or toxins. They become more and more concentrated. But do you think poisons become concentrated when we move up a food chain, going towards the larger animals, or do you think they become concentrated as we go down a food chain? It turns out that they become more concentrated as we go up the food chain. And the reason is because creatures, as they get larger, are eating larger and larger numbers of the smaller creature. So they're getting more and more of the toxins from those creatures. Okay, so that's what biomagnification is. So we have a question that kind of taps into that. So biomagnification, as we said, occurs within food chains as toxins become more and more concentrated as you move up the food chain. Let's see what this question says. It says, based on the graph showing the concentration of toxins found in the bodies of 4 members of a food chain, arrange the members to show what their order must be in the food chain. And the question says, arrange the members so that 1 is the member at the bottom of the food chain, and 4 is the member at the top of the food chain. Okay, so we know at the bottom, the concentration of toxin is going to be lower. So this creature here, phytoplankton, is going to be at the bottom of the food chain. Krill is going to be a little higher, so we're going to give it number 2. Herring's going to be higher still. It's going to be number 3. And puffin is going to be the highest at the top of the food chain. That's number 4. So what we need to do now is, again, write out what the numbers are. So we can see that herring was number 3. So first number we're going to write is 3. Puffin was number 4. Phytoplankton is 1. And krill is 2. So we're going to write those out, and that's going to be our 4-digit number. Okay, So moving along, we're going to talk at this point about substances that cause air pollution. We're going to talk about carbon dioxide and sulfur dioxide. Okay, so carbon dioxide, CO2, where does it come from? Well, it comes from cars as they're burning fossil fuels as they drive along. And SO2, or sulfur dioxide, mainly comes from coal generating stations, which we have a lot of in Alberta. Now the problem with sulfur dioxide is going to be different than the problem with carbon dioxide. What carbon dioxide does is it acts as a layer in our atmosphere, and it traps heat inside our atmosphere, and this is called the greenhouse effect. Okay, so light from the sun comes through our atmosphere, hits objects on the earth, it's changed into heat. And when it's changed into heat, the heat is not able to get back out of that layer, past that layer. And so the heat is trapped, and as a result, we have the greenhouse effect. And over time, the whole Earth could possibly be warming up some, and that's what we call global warming. So carbon dioxide is associated with greenhouse effect and global warming. Okay, so we've got some questions based on this. What is the problem with too much carbon dioxide gas in the atmosphere? Okay, we know it traps in heat. This is the greenhouse effect. Over time, this leads to global warming, or could. Where does this gas come from? Again, carbon dioxide primarily comes from burning fossil fuels, like gas. All right, question C, what's the problem with too much sulfur dioxide? Well, that combines with water in the air to produce acid rain. And where does that gas come from? Again, it comes from coal generating stations, which we have a lot of in Alberta. Okay, now remember, when you have really polluted water, what shows you that it's polluted is it looks cloudy. And another word for cloudy is turbid. The reason why it's cloudy is it has suspended solids in it. All right, now, very few fish and other creatures can live in acidic conditions. Also, very few creatures can live in conditions where the amount of oxygen dissolved in the water is low, or where there's lots of pollution. So in other words, fish like no pollution, they like a neutral pH, neither acidic nor basic, and they like high amounts of dissolved oxygen. So the question here says, which of the following conditions would lead to the greatest diversity of species? Okay, so diversity means a large amount of species, or large number of species. So what kind of pH do they like? Well, they like a neutral pH. So the answer is going to be either C or D at this point. Now, are they going to like cloudy water or clear water? Well, cloudy water is polluted, clear is not. So far it sounds like answer D. And again, they want high dissolved oxygen levels because, of course, many creatures need oxygen. So the answer here would be D. Next question says, here's some information collected from water samples. Says the concentration of suspended solids in milligrams per liter. At which of the sites is the water the most turbid? Okay, so in other words, when we talk about turbidity, that's pollution. And so when you have a high amount of suspended solids, that's going to be the most polluted. So we're looking for the largest number. So it's the greatest amount of weight of solid in a certain amount of volume. So that's going to be 54. Okay. Sorry, excuse me. We're going to talk at this point then about something called LD50 value. And remember, the smaller the LD50 value, the more poisonous the substance is. So again, LD50, the 50 part of the LD50 name, stands for 50% of the creatures being killed if you have a certain amount of a substance, of a toxin or poison. So what you normally do is you would take that number and you would multiply by the weight of the creature. So remember, the lower the LD50 value, the more toxic a substance is. So here's two LD50 values for two different substances. These here are different poisons that can kill rats. Okay, so we got some questions here. So again, what's the meaning of an LD50 value? Well, it means lethal dose killing 50% of the creatures. And part B says, which of the rat poisons would be most toxic and why? Well, the lower the LD50 value, the more toxic. So this one here is going to be more toxic. And the reason is it's got a lower LD50 value. It takes less of this substance to kill the same number of creatures. All right. So we're just going to touch on briefly some stuff about reading graphs. It's important to be able to interpret graphs and tell what they're telling us. So both these graphs are telling us that as time gets larger, speed is also getting larger. You can see that as time is getting greater, speed is getting greater. It's getting higher. But with this graph, speed is getting greater and greater over time. This is kind of like constant speed. This is kind of like acceleration. All right. So again, both these graphs are showing speed gets larger as time gets larger. Okay, so this graph here is showing a relationship between the amount of dissolved oxygen and the variety of organisms in a body of water. From this graph, you infer that the variety of organisms will, okay, do you think the variety is going to increase as the amount of dissolved oxygen will decrease? Or do you think the variety will decrease as the amount of oxygen will increase? Or will the variety increase as the oxygen increases? Well, as we looked at those two graphs before, we saw that both of them were showing that one thing was increasing as another thing was increasing. Except in this situation, things are increasing more and more. So the variety is increasing more and more as the amount of dissolved oxygen is increasing. All right. Now, we talked about four major substances that are in an environment that make up living things: proteins, carbohydrates, lipids, and nucleic acids. So let's just kind of talk a little bit more about each one of them and what they're composed of. Proteins are made of many amino acids joined together. Carbohydrates are made of many sugars joined together. Now, lipids are just another name for fats or oils. And nucleic acids, those are either DNA or RNA. Okay, so as we've looked at already, if we join three amino acids together, or many of them together, that forms a protein. Many sugars together form a carbohydrate. And some examples of carbohydrates are starch and cellulose. Starch is found in animals, cellulose is found in plants. So again, starch is just many glucose units put together. Okay, so let's kind of test our knowledge a bit. It says, explain how proteins and carbohydrates are similar in how they are made up. Well, we saw that both of them are made up of many subunits joined together. Proteins are many amino acids joined together. Carbohydrates are many sugars, or glucose, joined together. So that's how they're similar. Okay, let's look at three different words that we looked at before: diffusion, active transport, and osmosis, and let's kind of compare them and contrast them. Well, diffusion is the movement of a substance from a high concentration area to a low concentration area. So here, if you focus on the green dots, there's a lot of them here, and they would bump into each other, and over time, they would spread out. But they're going to move from this area where they're in high concentration to this area where there's fewer of them. And it happens automatically, diffusion does. We don't need any energy to make it happen, because things just bump into each other naturally. On the other hand, active transport does require energy, because you go from where there's few particles to where there's many particles. It doesn't happen naturally. You've got to add some energy to make it happen. And again, you're going the opposite direction here. You're going from a low concentration to a high concentration. Okay, now we're going to compare osmosis and diffusion. So osmosis is like diffusion. It's kind of a special type of diffusion, but it's different in two ways. With osmosis, it's water particles that are moving from one place to another, whereas diffusion is just any old particle. The other difference is that diffusion always involves a semi-permeable membrane. Why is it semi-permeable? Because it lets some things through and keeps other things out. Okay, so the question here says, explain how diffusion and active transport are different. Well, they're different in two ways. First of all, diffusion is going from an area of high concentration to an area of low concentration. Active transport is going from a low concentration to a high one. And again, diffusion happens automatically because all those particles close together just bump into each other and spread out. Whereas active transport does not happen automatically. It requires the addition of energy. Part B says, how are diffusion and osmosis different? Well, osmosis is like a type of diffusion. Particles are spreading out, but it involves water as the particles that are spreading out. The other important thing is that, remember, osmosis had that semi-permeable membrane that keeps some particles in and only allows other particles through. Okay, now we're going to talk about, if you've got some sort of pollution, solid pollution, how are you going to break it down and get rid of it? Well, there's 4 different ways you can do it. The first one is photolysis. Okay, photo means light, lysis means splitting. So basically, light can be used to split down substances, to break them down, and make the substance less polluting. Phytoremediation is another form of fixing up an area that's polluted. Phyto means plants, so what you can do is you can put some plants in an area. Those plants are going to take up the polluting substance through their roots, and then all you need to do is get rid of the plants, and you get rid of the pollution. Then there's chemical decomposition. Sometimes you can use a chemical and it'll break down some sort of substance. For instance, when you've got an oil spill like we had in the last year, you can use a chemical that will break down that oil because it's going to decompose or break down the substance. Now, biodegradation is another way of breaking down a substance. Remember, bio always means living, so it's using living things to break down substances. So you're using microorganisms or tiny creatures, like a bacteria, to break down some sort of substance that you want to break down. Okay, so our last question, we're going to explain what each one of these means. Photolysis, again, photo is light, lysis is splitting, so you're using light to split some sort of substance or break it down. Chemical decomposition, using some sort of chemical to break down a polluting substance. Biodegradation, bio is a living thing, so you're using like a bacteria or some sort of substance like that to break down a substance that you want to break down. All right, guys, thanks for listening. This is the end of the recording now, and so I want to encourage you again, practice all these questions till you can get them. Practice all the questions on the review, which is the second part of Lesson 12 and Lesson 13. If you can know all this material, be able to answer all these questions on this recording, plus Lessons 12 and 13, you should do well in the test, which is again going to be on December 2nd. So please study hard, and if you have any questions about anything, please let me know. Again, thanks for listening, guys. Have an awesome day. Take care and God bless.